Hi, my name's Taylor Darby, and I'm a first-year PhD student at San Diego State, rotating in the Ryester lab. In the Ryester lab, we study host pathogen interaction, and we focus on Trichomonas vaginalis. T. vaginalis is a eukaryotic, unicellular, flagellated parasite, and it's the most common non-viral STI in the world. Pathogens utilize multiple mechanisms of pathogenesis, but my project focuses on colonization and host immune response. To establish infection, T. vaginalis must first combat the native vaginal bacterial community, and a healthy vaginal microbiota is dominated by lactobacillus. T. vaginalis infection, so here our little lactobacillus is happy because we like lactobacillus. And with T. vaginalis infection, T. vaginalis infection is associated with low populations of Lactobacillus crispatus. So what we want to know is what comes first? Is there already a low population of Lactobacillus crispatus priming the conditions for T. vaginalis infection, or is T. vaginalis directly killing Lactobacillus crispatus, either by secreting antimicrobial properties or directly phagocytosing the Lactobacillus crispatus? So what we've found through immunofluorescence microscopy is the lactobacillus are directly binding to the parasite and surrounding the parasite, immobilizing it. And the parasite is also uptaking the L. crispatus. So what we want to do is verify these findings via quantitative microscopy to better understand T. vaginalis infection and the vaginal microbiota. Previous studies in our lab have found that macrophage exposed to T. vaginalis increase IL-1 beta secretion and Gasdermin D pore formation. This pathway involves cellular recognition of the parasite, and then via NLRP3 inflammasome signaling, increased production of active caspase-1, which then cleaves IL-1 beta into its bioactive form. And then bioactive IL-1 beta is secreted from the cell. Caspase-1 also cleaves Gasdermin D, and then the N-terminus of Gasdermin D oligomerizes in the plasma membrane and forms pores. These proteins can also escape through these pores. These pores also cause osmotic dysregulation, cell swelling, and pyroptosis, or cell death. What I want to know is what other cell types in the female reproductive system utilize this pathway when exposed to T. vaginalis. What I found is when inhibiting this pathway, placental cells decrease cytolysis when exposed to T. vaginalis. This indicates that plasma cells, or placental cells, utilize this pathway when exposed to T. vaginalis. I also want to look into whether vaginal cells, ectocervical, and endocervical cells utilize this pathway. Next, I want to look into what other protein secretion is dysregulated by these pore formations. Now we know that neutrophil infiltration increases with T. vaginalis infection. What I want to know is what other cell types are recruited to the site of infection due to the pore formation and the dysregulation of these secreted proteins. So this will help us better understand the host immune response to T. vaginalis infection and T. vaginalis colonization so that we can better understand how Trichomonas vaginalis causes disease. Thank you.